fundamental reason why the cancer arises in the first place. And in the specific case of breast cancer that I want to talk about today, breast cancer cases can roughly be classified into 3 categories. And shown here are, again, schematics of cancer cells. So this is the cell body, and the red circle within is the nucleus. So about 60% of breast cancer patients have so-called ER positive cancer cases. ER is short for estrogen receptor. And it's one type of driver that drives breast cancer proliferation. And there's another category of patients that account for about 20% of new cases. It's called HER2 positive breast cancer. Now, as I'll talk about in the next slide in more detail, HER2 is a protein that's sitting on the surface of cells. And the rest of the breast cancer cases are, to the best of knowledge, it's a heterogeneous group. It's, the mechanisms of which are relatively ill-defined. So, and that accounts for the other 20% of the cases. And I should point out that this category, which we will focus on today, HER2 positive breast cancer, is a particularly aggressive subtype of breast cancer, and women tend to fare poorly in terms of their survival after diagnosis.
Could you say again what that little black thing on HER2?
This thing.
That makes it HER2?
No, that's the protein HER2. Yeah, it's the I hope I'll.
It's on the cell.
Yes, it's sitting on the cell membrane. And very good question to talk about. Yeah. So what is HER2? HER2 is a protein that's called a receptor that sits on the cell membrane. Physiologically, it functions as a green light signal that's pro-cell division, pro-proliferation. So it gives the cell a positive signal to actively grow and divide. And it's not a bad protein in the sense that every one of us have that protein. When it's active under proper regulation, it's doing its normal job in the human cell. So, yeah, it sends this, you can think of this as a green light. It sends a growth signal into the cell. However, in the cancer cells of HER2 positive breast cancer patients, what you see is this. So there's abnormally high production of HER2 protein on the cell. So essentially you have too much growth signal. So I mean, as you can imagine, too much growth signal disrupts the balance, and therefore precipitates cancer development. So now we know the cause of this subtype of breast cancer. We can properly design a therapy that's particularly effective at combating those type of cancers. Now, fortunately, we have a drug, Herceptin. So what Herceptin does, Herceptin is a therapeutic agent that can physically bind to HER2 proteins that's expressed in abundance on the surface of cancer cells. And by binding to it, it inactivates it. Therefore, you cut off this access of growth signal that would normally have made its way into the nucleus of the cell and encourage the cell to divide and to grow. Right. So it's all good that we have this drug and we can use that to properly shut down access HER2 signaling that leads to cancer. But as you can recall, only 20% of the women who have breast cancer have HER2 positive breast cancer. So how would an oncologist figure out whom to give this drug to? Because if we give this drug to everyone, 4 out of 5 men will not respond to this drug because they do not have HER2 positive breast cancer. That's not the reason why they have cancer. So clearly that drug will not be effective. And fortunately, now we have this clinical diagnosis kit called, basically it tests whether a particular patient have HER2 positive or HER2 negative breast cancer. And how this kit works is as illustrated in those 2 figures. Those were tissue biopsies taken from the tumor after the patient underwent surgery to remove their tumors. And the green dots are basically you're labeling the chromosome, part of the chromosome where the HER2 resides. So the green dot indicates that HER2 is normal, and the red dots indicate that HER2 is have underwent expansion, and so which will lead to overproduction of the HER2 protein. Right. So now we have this drug. We have this way of telling whether a patient have HER2 positive breast cancer, and we want to see if this drug is effective at helping those women with HER2 plus breast cancer, and it does. So this is taken from a primary literature on one of the clinical trials that used HER2, and I would want you to focus on two curves, the one in red and the one in orange. So, and this is a survival chart. It basically documents how well the patient does from a given set point on how well they survive after their cancer diagnosis. On x-axis is the number of years from the beginning of the study, and on the y-axis is the percent of patients that's still living. So we start at 100%, and unfortunately, some patients deceased after the beginning of the study. And you see that for women with HER2 plus breast cancer not treated by this drug, there's a much sharper or much higher mortality rate compared to HER2 positive breast cancer women treated with this drug. So it's a very effective drug.
Why are the untreated population such a high and treated population surviving? 60%.
Some will survive for reasons other than
HR2 untreated. Other
therapies might work
Other
for them.
therapy, okay.
Yeah, just not this particular drug.
So if they were untreated, that would plummet too.
Yeah.
And also keep in mind the point that Monica made is, although you have a particular gene that may cause cancer, but that doesn't mean that you will have 100% chance to get cancer. Right?
Are these patients already with cancer?
No, I mean, those patients are HER2 positive. But that doesn't mean that all of them will die because...
There are other effects of the environment that could really influence how long you live, like your nutrition.
They all have breast cancer.
Yes, they do.
Yes, they're all breast cancer patients. But the point of the chart is that if you have the specific kind of breast cancer, this medication helps you live longer. But if you don't have this particular kind of cancer, you waste a lot of time and money.
The HER2 untreated, whatever else they were treated would have to be consistent, wouldn't it, for the data to mean it? Okay.
The difference between basically the red one and the purple one, maybe these patients are a combination of treatment. The difference is the
So-called
absence
standard
of
treatment.
You're right, yeah. Yeah, because you're only treating patients with cancer with different condition drugs.
That's a very good question. And it's great that not only it's pharmacogenetics making its way into the field of breast cancer care, it really has flourished in many different fields of medicine. For example, therapeutic principles based on pharmacogenetics has been effectively used to manage medical conditions, other medical conditions such as leukemia. It's leukemia, colon cancer, cardiovascular diseases, HIV, pain, and other neurological disorders. Now, in summary, pharmacogenetics, in essence, is to give the right drug to the right patient based on the genetic information of individual patients. And we walked through a successful case of applying pharmacogenetics in managing breast cancer using the drug Herceptin as an example. And pharmacogenetics is really making its way in other clinical fields other than breast cancer care. And one future aspect of keep improving this concept of this practice of pharmacogenetics is to move from pharmacogenetics to pharmacogenomics. As Monica said, we have 25,000 genes in our body. So diseases or patient profiles that can be classified by a single gene is really a rare thing as opposed to a common thing. So many diseases are defined by a particular genomic profile of many genes changing in a certain manner. So scientists have started to look at more than one gene at a time and classify patients that way. And there's great hope that we'll do a lot better things using pharmacogenomics. Right, so now Eric will give the next phase of this talk, basically focusing on stem cells. So pharmacogenetics, even including pharmacogenomics, as great as they are, have limitations. For example, for a lot of complex diseases, a single therapeutic agent may not achieve the total therapeutic effect. We may want something more sophisticated, a therapeutic tool that can target multiple things in a more dynamic manner. So what would that be? I mean, how about tweaking some of your own cells to do the therapy? Because as Monica mentioned, we have this great hierarchy of biological organization from DNA molecules to different molecules group and form into cells. So cells is a lot more sophisticated than a single chemical or a single protein. So Eric will tell you more next. Thanks.